next speaker then is Zvi Mendel, talking about chemical control trials for fornicatus.
Okay, ladies and gentlemen, I would like to share with you our not very successful experience with chemical control of the beetle, and later on Stanley will tell you something about the chemical control of the fungus. Actually, the avocado production in Israel is very similar to organic cultivation. Growers don't use any insecticides or fungicides in these plantations. So this is our advantage when we export the avocado to the European market. So when we need to think about chemical control, it was a bit unheard. When we learned about the problem and we begin to study the options, we realized that biological control and using pheromone is not an option, at least for the short run. So we need to consider something else. And the first something else was an idea to cover the tree with lime. So maybe this will deter the beetle and we will be able to protect the trees. So we did some lab experiment. As you see, we covered some of the branches with lime, other not. We put this tube on the branches. We put 2 beetles inside and see what is going on. And as you can see, you see the frost coming out in the control and protected the branches, so actually it didn't work for us. And we did the same thing with young samplings, like this one. Part of it was covered with lime, the other one is not. You look closely and you see that in both cases the lime was not able to protect the penetration of the beetle inside the stem. So we had to start to think about the chemical control options, and we tried different things. So what you can see here is a long list of chemicals, both insecticides and fungicide, with the attempt to do something in order to control the beetle. We excluded the nasty stuff like the organophosphates and the carbamates, and we stick to mainly to systemic insecticide because we thought that systemic insecticide is our best bet. And then you can, then you see there are different groups of different classes of insecticides. And first of all, we test most of them in the lab, and later on, the successful ones were also tested in the field. So at first, we start with the systemic insecticides, and we develop a simple laboratory test in order to see whether the insecticides are effective or not, with the idea that we should try to kill the larvae. So the idea is, if we wish to kill the larvae, we need to use something that it will pick up by the fungus inside the trees, since the larvae are feeding on the fungus. So the idea that we will inject the trees, the fungus will pick up the insecticide, and the larvae will feed on the toxic fungus. So we use this system that I showed you, Stanley showed you earlier, and the process was this. In the lab of Stanley, we prepare the PDA with the tested insecticides, and after a week, when we see the development of the fungus, we expose them to larvae of different ages, and you see the larvae feeding on the fungus again, And here are the results. So, for example, here you can see the effect of the insecticides on the performance or the mortality of the larvae. And you can see here that here 3 compounds show where seems to be promising. One of the imidacloprid, Confidur, this compound with this complicated name, which is a new compound. However, this compound is also coupled with a pyrethroid, which actually did the job and not this new compound. And then there's another neonicotinoids. All 3 of them managed to kill all the larvae within very short time. About 6 days was enough in order to, or in this case, 9 days were enough to kill all the larvae. One insecticide which was a bit of disappointment was the azadirachtin. Azadirachtin is very effective insecticide against beetles, and somehow it didn't do, it was not very effective. We use azadirachtin in 2 ways. We incorporated the insecticide into the PDA, as we did with the other compounds. But because when we prepare the PDA, you have to warm it up. So we were afraid that maybe this heating destroyed the azadirachtin, which is the case. So in those cases where we are afraid that the heat will destroy the compound, we also, besides incorporating the insecticide into the PDA, we also spread it after the PDA reached the room temperature. But again, you see we have here 3 insecticides that seems to be very promising in the lab. This is another group of insecticide that we tested. Again, some of them are neonicotinoids, some other belong to some new group of systemic insecticides, and in this case, only the neonicotinoid, this one, thiometaxam, did relatively good job, and after 11 days, I want to emphasize that most of the larvae were killed by the insecticide. And this is the last one in this series. Again, here we tested 2 interesting compounds. One of them is spirotetramat, again, a new compound of Bayer, which a very interesting behavior because this insecticide is traveling along the plant from the roots to the top or from the top to the roots. Again, sensitive to heat. It was not very effective. And the other one was emamectin benzoate, which is also an interesting insecticide, mainly because it's considered safe. And you can use it as a systemic insecticide if you inject it to the stem. And again, this seems to be very promising. You see that we didn't see any mortality in our control trials. So we could move to, first of all, I would like to show you some of the photo, how these things look like in the lab. As here you can see the effect of the imidacloprid or the emamectin benzoate with a close look. Again, azadirachtin, it's a bit difficult to see that, but let me show you some other images. Here, for example, you can see the effect of the Confidur, imidacloprid. You see the dead larvae, or from a closer look, this is a very typical of larvae that were feeding on PDA with Confidur. Or here, the effect of emamectin benzoate on the larvae, a closer look, and you see the dead larvae. And on the other hand, you see the larvae look very nice with their typical feeding position in the control replicates. Or here, you see the larvae, they're actually, eventually they will clean everything on the plate. Then we move to the field trials. What you see here is a fleet of cars belong to the different chemical companies that took part in the in those experiments, but they were very happy to take part because they dreamed they will sell a lot of stuff. And again, very many people together in one of the plantations. And of course, our first option was to try stem injection. That seems to us the most promising or safe strategy to introduce the chemical into the tree. Again, well, it looked nice, very tedious. However, we thought that that might be done under certain circumstances. However, the results were not very good. Here, for example, you can see the list of results with no much differences between the control and here the untreated trees and the different compound we tested in one of the plantations. So you see several insecticides were tested. Some of them were tested by soil application. So we introduced the compound through the drip irrigation system. In other cases, we simply, this one was not a stem injection, but spray cover of the stem and the branches. And you look at the results, we didn't see anything promise. In each case, we also took some branch samples in order to see what's going on inside the branch. I want to mention that our major measure that I'm going to show you is the number of penetration holes. So we actually count the number of penetration holes on every tree, and that was our major measure to figure out whether the application of the insecticides was effective or not. Sometimes it's a bit difficult to do that because you can figure out that counting all those in penetration spots is not very easy. And sometimes we tested the effect of the insecticides by simply spraying branches using those Eppendorf tubes and expose the beetles in certain different periods after we actually treated the branches, and we measured the mortality rates in order to have some idea how much the insecticide actually remains effective. With the stem injection trial, we faced some problems because not all the compounds that we tested really absorbed by the tree. In this, for example, what happened here after we conducted the stem injection, and you see that most of the material actually remain outside. Okay, and then of course after we did parallel to the examination of the condition of the tree, we took some branch samples. Here you can see we work in the garage of one of the kibbutzim in the area in order to see what is going on inside. This is Alex trying to see what inside. And we open each one of these branches, and we gave some estimates to the level of infestation and the spread on fungus. And in all cases, this is the control, this is the confidure, for example, and in all cases, what we could see that we were unable to reduce the level of injury caused by the beetle. Again, this is in one of the neonicotinoids, which seems to work in the lab, but unfortunately, in field test, it was not very effective. This is another experiment we conducted. Here you see the comparison between stem injection, soil application, and spray cover on the stem and the branches. And again, we didn't see in this particular case any effect of each of the treatment. This is another experiment done in a private plantation, a small one with mature trees. First we use the pyrethroids. So we covered, this lady had two small plantations, one the southern plot and the northern plot. One of the plots we treated with bifenthrin, the other one was treated with another pyrethroid, and again, we couldn't see any differences between control and untreated tree. Since the lady was generous enough to let us do whatever we wish to do in her plantation, we then use a confidure in order to try to prevent further infestation of her trees. And again, what you can see here, we compare the situation of the number of trees which are affected, and these are, here it shows the group of trees according to the spot density, and you can see there is no real difference between control trees and treated trees. So actually the use of confidure through the irrigation system was not effective at all. Again, another plantation, this time is reed. Again, we tested several compounds, those that seems to be effective in the lab. There are some insecticides, and we also tested some fungicides. Stelly going to talk about them a bit later. In only one case, we could see something, but not in this particular plantation. So you can see that between those two dates, we could observe an increase in the number of penetration spots, and none of these chemicals really did something useful. This is another plantation, young trees, and this is a new systemic compound, which was not very promising in the lab, but we used it anyway as control. A fungicide and confidure. Only in case of confidure, we got some good statistics that the confidure did something to slow the accumulation of penetration holes on the stems. But in all other cases, we didn't see much of a difference between the two dates. I think this is one of the last slides. Again, another plantation. Again, 15-old plantation. And you can see, again, we tested different compounds, both fungicide and insecticides by stem injection and by soil application. And only in the case of emamectin benzoate, here, we managed somehow to slow or to stop the accumulation of penetration spots on those trees. In all other cases, we couldn't see any effect of the treatment. And this is the last slide. In this case, we sprayed branches in order to protect them from further infestation. And what we could see here, that the spray with one of the neonicotinoids give us some nice results here compared with what happened with the controls. So we concluded that spraying the branches may be a solution, or spraying the infested spot might be a solution in order to prevent from the beetle to come out, or to protect the branches. Thank you.
Is there a registration for emamectin benzoate for avocado?
There is no registration for anything yet.
Okay. Yeah, well, I'm asking because the company doesn't seem to be interested in doing that.
Well, the fact is that emamectin benzoate as a spray cover, it's a lost case because emamectin tend to disintegrate very fast. However, as a systemic insecticide, is extremely good, usually against other pests like defoliator, for example, or trips, but not in this case. Yes, please.
Did you test any surfactants to help the material maybe move into the trunk?
Again, please.
Surfactants, like Solette or something like that, a material that would allow the product to move into the trunk easily?
I see, no.
Zvi,
No, we use commercial compounds.
Zvi, most of the compounds you tested were insecticides, is that correct?
Sorry, I can't hear
Most
you.
of the compounds you tested were insecticides, is that right? They were insecticides. You tested insecticides mainly for control of the beetle?
Yes.
Okay, would a better strategy have been to try a fungicide to kill the food source for the larvae? So if they were in the tree, they would have starved to death?
Some of these were, some of the compounds that we tested were systemic fungicide. However, I'm not sure they are very good, and I believe that Stanley will refer about it a bit later.
From the last set of insecticides, the ones that gave good results, do you have an idea of how long they can protect the tree, their persistence in field conditions?
Well, according to the results I've just showed, it seems that they are not effective at all under field conditions.
But in the last one, you showed some...
Yes, in the fourth, we don't know yet. Mm-hmm. So we still study some of these compounds to find out how long they will last. Because the idea is to figure out what kind of insecticide you would like to use, and what is the purpose. So one purpose may be to protect the branch from being colonized by the beetle. Another idea is to try to use insecticides in order to kill the emerging beetles. And that might be even more important in some cases, especially if you think about prevention. So the idea is that extensive monitoring will allow you to see the first penetration spots, because these spots develop very quickly. Couple of days, or 3 or 4 days after the penetration of the beetle. So if you discover those early spots, you may spray the branch and then prevent the beetle from coming out or further colonization on this particular spot.